The microbiology lab is an integral part of patient care here at Loyola University Hospital. From diagnosis to treatment, stewardship, and infection control, the microbiology lab is involved in the day-to-day running of this large institution. Like any processing situation, it is exceedingly important to understand the information the micro lab provides us, and as physicians, make educated decisions about patient care, incorporating this information appropriately. While the basic job of the microbiology laboratory has remained essentially the same, that is, identification of organisms and their susceptibilities, some of the work that these scientists perform has changed, often making our job as physicians somewhat easier. Today, we will tour the microbiology laboratory here at Loyola, that nebulous place in the basement where samples disappear and we await results of their testing. You may have heard of such instruments as BACTEC, BioFire, MALDI-TOF, Microscan, and Cepheid, without really understanding what they are or what they do. This trip to the microlab will introduce you to these state-of-the-art instruments that are key to our day-to-day decisions in patient care in the 21st century. As with any effective system, garbage in equals garbage out, so we will start with sample collection because proper sample collection is key. Keep in mind that in addition to drawing blood cultures prior to administration of antibiotics, there are other important factors that can affect sensitivity of blood cultures. It is important to obtain a sufficient amount of blood in the blood culture bottle. In reality, less than 50% of submitted blood culture bottles are actually ideal with respect to quantity. For every 1 ml missing, there is a 3% decrease in sensitivity. For bottles only half full, the sensitivity decreases to 70%. So basically, patients could have bacteremia that we do not detect because we did not optimize our blood cultures. The right amount should be 8 to 10 cc's full. So we have Erin and Lauren here in specimen receiving, and I'd like to ask you a couple questions about what happens when the specimens come in. What types of specimens do you get? How do they arrive? What are some issues that happen that you have to deal with in terms of processing the specimen or having difficulty processing the specimen?
Okay, so to start, the way we get things, most of them come through that pneumatic tube station that's over there. That's constant all day long. There are a few things that do come through the window, so the irretrievable specimens, so your OR specimens, your spinal fluids, all those kind of things come through the window here. And no matter where it comes from, the first thing we're going to do is check it, make sure it's not mislabeled, make sure it's in the correct container, and then we're going to receive it in our computer system.
Okay. Mm-hmm. And then where do you go from there?
From there, we kind of decide what takes priority, and so that would be stat specimens. So mainly those are ER specimens, and we can separate those from the rest because they are in red stat bags. So we pull those first. We usually get buckets at a time depending on the time of day. So we'll pull those, we'll run those, as well as any spinal fluids we might get that come through the window. Those take priority over everything. So we'll get those set up right away. Next would just be PCR in general. We have about a 2-hour turnaround time for all of our PCRs. So as soon as then that 2-hour turnaround time is from the second we receive it, not from the time it's collected and they said they sent it to us. An issue we have is that most floors send everything to chemistry first, so core lab, the main lab. However, chemistry batches everything until they have a full bin and then they send it to us. So sometimes doctors get upset that they said they sent something a while ago, but we haven't received it, and that's because chemistry still has it. We actually don't have it in our lab. So they either have to be lenient about that, or if they want to send it directly to us, we have our own tube station and they can do that. It's just that they know that one better.
Oh, so they don't actually send it to micro?
Sometimes.
They send it all to chemistry?
But for the most part, yes.
So that's a problem.
Yes.
So that can decrease our wait time by just taking that extra step and sending one to chemistry and one
to micro.
Okay, wonderful.
Yeah.
Good to know.
Yeah.
Any other issues? What about labeling? Any concerns with labeling or any problems with that?
So blood bottles are a big thing. Depending on where you put the label, it can make it like easier or harder for us. So there's these spots on these bottles. When you put the patient label on it, it will look like this. It doesn't have any patient info on it right now. But you want to put it here where it's not covering up this barcode or this sticker, because we're going to need to remove this sticker later. This comes off, this little part right here, and we need that for stuff.
Oh.
So if you put this label, the patient label, over this, that complicates things for us.
Oh, so then what do you do?
So we have labels in the back. We call them dummy labels. They're not associated to any bottle or patient ID. And so when that happens, we have to manually scan it in and then type in the code we want it to say, the time, and then the amount of days that it's supposed to be held. So these bottles are pre-programmed, and so it knows how long it's supposed to hold that bottle. It has a container ID to go with it. As soon as we put one of those labels on, it's kind of blank. And if we accidentally put something in wrong, that could be an issue.
Oh, wow. And these are the Bactec bottles?
Correct.
Yes.
Yeah, these
Okay,
are
we're going to
blood
talk about the
filters.
Bactec machine a little bit later.
Yeah.
So yeah, and that seems to increase your work, and it then will extend the time for our blood culture results.
Yes, and it could theoretically cause issues with mislabeling or perhaps like the wrong patient or the wrong bottle number, and you just don't want to get into any of those type of problems.
So this is really important for patient safety and patient care.
Yeah.
Right, and then also along those lines, with these labels, when you're labeling them, make sure you put like the date and the time that you're collecting it, so that we're aware that it's the correct date and time, not just what printed out on the label. It might be different.
Oh, okay.
And then the site also that it's being collected from. Sometimes it's not the same site that they ordered it as in the computer originally, so that needs to be noted as well.
Okay.
Sometimes, especially, and this does happen a lot with our aerobic, anaerobic cultures, when they'll be ordered just very, very vaguely, just wounds. But we need to know specifically where that's coming from. We have lots of normal flora all over our body. It helps us to determine what we work up on the benches if we know exactly where it's collected from.
Okay.
Or sometimes it'll say other, and we have no idea what that means. So we have to go tracking down who collected it, what they actually collected. And it's just much easier right from the get-go. It said what we needed.
Okay. Wow. That's that could be a problem. Let's see, any other issues with any specimens? Let's say biopsies or tissue specimens.
Yeah, so with tissues and fluids, it's always best, if you want a culture done on it, to get the actual tissue and the actual fluid. It's not good for us when we get it on a swab. It's not holding enough of the actual sample. So if at all possible, always we want the fluid or the tissue. If you have to put it on a swab, we don't have any here. The glass portical tubes are the tubes that you need for the aerobic and the anaerobic. If you just put it on a regular swab like this,
And that's just the fluid, right? Just fluid.
if you're using a swab for a fluid, if you have to, and you put it in one of these tubes, this is only going to get an aerobic culture.
That's
the only thing
only-
that it's going to
okay.
sustain. So a lot of times we'll get these that'll have the aerobic and the anaerobic ordered on it, and we have to cancel it. We'll run the aerobic, but that's all we can do.
Why
so
is
if
that?
this doesn't sustain the anaerobic.
The
So
anaerobic.
the swabs don't allow some things to grow that. Is
Correct.
that right?
Yes, the anaerobes will die off immediately, very quickly. So that's why those other portical tubes she's talking about, they have a gel medium in the bottom. Okay. And so once you put the swabs in there, it takes a few minutes to equilibrate, and then
Here, this one?
yes, and then so it's this one's a little bloody, but it will turn pink immediately, and once it sits for about 15 minutes, it will equilibrate back to this clear color gel, and that's how we know it's in an anaerobic environment.
Okay, and that's dark.
Yes, it's a black cap, clear tube, glass with this clear gel at
And
the
that's
bottom.
for anaerobes.
Yes, but this is kind of last case scenario. We would much prefer the fluid, even if it's just a drop, even if it's just a biopsy of tissue, we would prefer any of that. Swabs, not only do they get stuck in this gel, we have about 7 plates we use for anaerobes, and if you have to plate all of those and a gram stain, you're using up the majority of whatever's on that swab.
Okay.
So it's better, it's much easier to recover things when you get the tissue.
Specimen gets left behind in the gel too. So it's never a good idea. I don't think it's acceptable to put, sometimes people will put fluid into this tube, like as a transport. tube
Oh, wow.
for fluid or for tissue. They'll like jam it down
I've
into the
seen
gel.
tissue jammed in
I mean, the opening for this tube is very small, and it's borderline impossible to get it up and out of the gel. Just put it in a sterile cup.
Okay, in a sterile cup. What about in a syringe?
Yes.
That's good.
Can you just like recap the syringe and then put it in a bag?
As long as the needle's been removed. If it's
Well,
not,
we wouldn't use a needle. No needle?
okay, yes, no needle.
no
So
needle.
there's some people taking like aspirates of things.
Gotcha,
Yes,
okay.
and so it'll come down and it'll just have the cap back on. But if we see the needle underneath it, we won't touch it. We won't accept it.
Needle has to be removed,
Yes.
and then you just cap the actual syringe,
Yep.
or you just put the plastic.
Yeah, and you can put the patient label right on there and send it down in a bag.
Okay.
Yes,
If
that is
it's-
preferred.
Yeah, if it's low volume, a low volume of an aspirate or a fluid, it's best to leave it in that syringe so that you're not transferring it to a cup and then some is left behind. And then sometimes it'll also like go up into like the, what's the top? I can't remember what it's called.
The actual
With that
syringe part,
plastic?
yeah.
Yeah, it'll like go up
Yeah.
into like the spiral part of the cap, and then we lose some of it too.
Oh, okay. Okay, that's good to know. Okay, wonderful. So aren't those the blood bottles that we saw in receiving?
Yes, they
In
are.
Specimen receiving? Okay, so what's going on here? What is this?
So this is our back tech instrument. Each one of these holds approximately 100 different vials. So she is scanning the patient identifier as well as the bottle identifier. So this is why it is so important that we didn't cover up those labels. We were
Okay.
talking about the labeling earlier. So she's scanning both of those in and then placing them in one of the little slots that it has there. And then based on the type of bottles, so we have these as well as our fungal and AFB bottles. Based on the type of bottle we scan, it automatically knows how long to hold the bottle. So the aerobic, anaerobic, those blue and the purple ones, it will hold for 5 days. So if it's negative, it'll hold it for 5 days. The fungal and the AFB will hold for 30 and 42 days respectively.
So they're held mix and match.
Yes, we can put them anywhere.
But when you scan it, it'll say this is fungal or this is
Yes,
AFB,
exactly.
so we have to hold it longer.
Yes.
Okay, all right. So what happens in here? What's going on with the bottles?
So this is a continuous read instrument. It checks every 10 minutes, I believe. So we actually don't really need to do any intervening until it goes positive. So there, as the organism grows inside the vial, it's producing CO2 and it mixes with the dye. And the more CO2 it releases, the higher of a threshold it reaches. And once it crosses this peak threshold, it creates like a fluorescent in the bottom of the bottle, and there's a sensor that is detected by the slot that we put it in. And so once that happens, this goes positive. And if you don't mind, I'm going to demonstrate what it looks and sounds like when one goes positive. So the first thing we'll see, we won't see all of these colors. It will turn red. Red means it's positive.
Okay.
Green means they're negative, and yellow means it's an issue with the instrument. So it'll go red, and we will have this noise go off.
Okay.
And so this happens all day. We're open 365, 24 hours a day. So it'll happen on all shifts, and we don't know how many or how often this is going to happen. So that's what it looks like. As soon as it goes positive, we have 2 hours, so similar to the 2-hour turnaround time up there, to make a gram stain and then decide what type of PCR it will get. And then after that, we have to call it up to the floor as a critical report. So we will be giving the doctor or the floor the gram stain result as well as a correlating PCR result.
Okay. So it pops up positive. You don't notify the doctor until you've gotten the PCR report.
Yes, yes. So the gram stain is 100% necessary. We won't just tell them that it was positive. We need to give them some sort of information. And now, since we are lucky enough to have the BioFire, it's kind of better and more beneficial to the patient if we can tell them what the organism is right off the bat. So then they can start treating based on that organism rather than just like a vague gram stain.
Okay. Okay. So why does this have a green and a red?
Okay, so in this particular cabinet, that just means we have one positive and one negative in there right now.
Oh, okay. Okay. So if it's positive, don't you take it out?
Yes.
Or, okay.
Yes, yeah.
So that one hasn't been taken out yet?
No, so that is an anonymous bottle that was for your guys' benefit.
Oh.
So that's yes. So, and as soon as we have one that is positive, it flashes red when we open it up so we know which one is alarming, and we will pull it. and then the rest of them go black. So now we know we have the one that it was correlating to. If I go back to the home screen, it will now flash the one that's negative. So all the rest of these, they're turned off because they're pending, and the green are available. So the flashing green is the negative one. So now, so pending are the ones, so when we scanned it in, it held it for 5 days. And so it's holding on to those for 5 days. So they've been negative thus far. So we keep all of the fungal bottles in here, just so we keep them all in one location and we know where they all are.
Okay, so yeah, they are.
Yes.
Now I see blue. Is that like fungal and then AFB or?
Those are just other kind of like studies.
So some extra.
Yeah.
Okay. Okay.
Yes. But the rest of our patient aerobic, anaerobic ones we'll keep in these.
Okay.
So it's just kind of a special drawer.
Okay, wonderful.
Yes.
Wonderful. Now, even though you're getting fungal blood cultures, Cryptococcus grows in regular blood cultures, right? Candida does.
Yes,
Yeah.
yes. So a lot of time people will try to order a fungal blood just for yeast. Yeast will grow very easily in our normal blood bottles. So there's really no need to order both if you're concerned about yeast.
Right, so it's more for molds, right?
Yes.
Like the fungal ones.
Uh-huh.
So more for like our cancer patients, bone marrow transplants, chemotherapy,
Yep.
or even our organ transplants, immunocompromised
Yes.
patients usually.
So revisiting a common thing that we see in the lab that we had mentioned earlier is that we do get patient barcodes that do cover up the bottle barcodes. Both barcodes are used when putting them on the instrument, so it really does help if this barcode on the bottle is still exposed. For example, they have a groove here with a laser that detects the barcode and will put all the information on the instrument. So we do need this exposed to be able to load the bottle, and it can determine what type it is, how long it should be incubated, and so on and so forth.
So this is the BioFire station, right?
Yes.
So what is BioFire?
BioFire is a company that runs PCR method testing to rapidly determine whether or not a patient might have various pathogens. We do different assays with BioFire, one being the respiratory panel. We have a stool PCR panel, a blood panel, a meningitis panel, and there are more things to come. So it's an up-and-coming, fairly new, good system.
Okay, so how many can you run at a time, or how often do you batch them, or how does that
Sure. So if you look actually here at our instrument, we have 10 slots. Technically we can run 10 at a time. Each slot is specific for one specimen at a time, and we try not to batch the specimens. This is a PCR method, meaning we can run this test and get the results in a little over an hour. So we want to utilize the technology, and we do run those as a stat priority.
I wanted to ask you, with this BioFire, can you run any of the several different types of specimens at the same time, or on this machine?
Sure, yes. Each slot can run any of the assays. The assays are specific to the barcode on the pouch, so it can read which assay we want when we scan our specimen prepared in the pouch. However, we do divide up each slot for specific assays if possible. For example, there are some slots that are specifically designated for the stool PCR panels only, some for the meningitis panel only. This is to minimize any risk of contamination and also to assure that each assay has a slot and can be run. Yeah.
And so you probably get a lot that are stat everything. We love that word.
Yes.
So how do you determine which ones to do first? Otherwise, the few that aren't stat might be sitting on the counter for a long time.
Sure. We have a priority, and we use our best judgment in every case. However, stats usually come from emergency departments, cancer centers, anything that is inpatient first. Then we'll do some of the outpatient specimens. Like I said, we try not to batch them, but when we get a couple stats at the same time, we'll wait till we all get them in and run them all at one time to be as efficient as we can with our turnaround times.
Okay, great. And so with the BioFire, I understand that you can test for many different organisms.
Sure.
But not everything.
Right. So each assay has a specific list of potential pathogens and targets that it does test for. It's a wide list and it's different for every assay. However, it is not completely comprehensive. There are also other specific PCR options. Usually those are send-out tests that are done in other reference labs.
Okay. So my understanding, though, also is that with the blood biofire,
Yes.
you can also detect sometimes certain organisms that have certain drug resistance, correct?
Sure. So one bit of technology that this instrument and this system offers is that they do detect genes that have resistance mechanisms in some organisms. Of course, this is always confirmed with other biochemical and culture method testing as well.
Okay, and that's very, very helpful for us.
Of course.
So would you be able to show us how
Sure.
you set up a sample?
Of course, I'd love to demonstrate for you. So to our right of the hood, which is difficult to see, we have the materials for the respiratory panels. We keep it separated from other assays such as the GI panel, and behind you would be the meningitis panel. We also do have blood panels by the respective blood bench. So we do have a respiratory panel sample here, which I will show you how to run. Next, we have our pouch. This is a closed system, vacuum sealed in this package. This was the barcode that I had previously mentioned. As you can see, it says respiratory panel. So once we scan this onto the instrument, it should verify that it is a respiratory panel sample we are running. When we are finished, it will pair up anything that was in the specimen against the listed targets that it can run.
To begin a film array run, the pouch is first placed into the loading block. Then, hydration solution is injected into the pouch through the blue inlet port on the right. The vacuum in the film array pouch automatically draws the correct volume, eliminating the need for precise measuring and pipetting. Next, sample is added to the film array sample buffer. This solution is then mixed and then injected into the film array pouch through the red inlet port located on the left side of the pouch. Again, the vacuum in the pouch automatically draws the required volume.
So we select the module and scan the barcode on the pouch. This will indicate which assay we are running. Next, we scan the patient sample. That way it correlates. We load it onto the instrument.
Here is how the biofire works.
Now let's take a look at what happens inside the pouch. First, the sample is moved into the lysis chamber where the film array physically lyses any cells and viruses through a process called bead beating. Ceramic beads are agitated at high speed to break open cells and viruses and release the nucleic acids. These nucleic acids are then bound by magnetic beads, which are then moved from the lysis chamber to the purification chamber. Here, a wash buffer removes any remaining cellular and viral debris. The film array activates a magnet outside the pouch, which holds the magnetic beads in place while the debris is washed away. Next, an elution buffer releases the purified nucleic acids from the magnetic beads. The magnetic beads are again magnetically secured while the nucleic acids are moved to the first stage PCR chamber. A reverse transcription step is performed to convert any target RNA into DNA. This is followed by a high-order multiplex PCR involving dozens of primer pairs. During this first stage PCR, many reactions simultaneously occur in this single large volume reaction. The products from the first stage PCR are then diluted to limit any remaining first stage PCR primers. Next, the diluted first stage PCR products are combined with a fresh master mix and then aliquoted to each well in the array. Each well of this array is pre-spotted with one pair of second stage PCR primers. These second stage PCR primers are designed to amplify sequences contained within the products from the first stage PCR. This eliminates non-specific products associated with traditional multiplex PCR, because the second stage primers specifically amplify only target DNA. A fluorescent double-stranded DNA binding dye is used to monitor each reaction. The second stage primers in each well are designed to detect one specific target. Organisms are identified based on which wells in the array are positive. The film array performs a melt to confirm the presence or absence of specific temperature signatures of the second stage PCR products.
From this point, we do get a printout of the specimen, the test, excuse me, and it has a list of targets that it does detect for here. These are all the targets that it detected for. In this particular sample, no targets were detected, as the results indicate. It tells you specifically which target for each of them also that it tested for.
Unfortunately, the PCR does have its limitations, and we still have to plate the organisms from culture, often for further identification and susceptibility testing. Fortunately, here at Loyola, we also have MALDI-TOF for further identification of not just blood, but other culture samples.
You work this MALDI-TOF, okay, and I'm sure a lot of people are asking, what the heck is a MALDI-TOF?
Sure.
What is it, and what do we use it for?
So MALDI-TOF stands for Matrix-Assisted Laser Desorption Ionization Time of Flight. So we refer to it as the MALDI-TOF, or MALDI for short. And essentially what it is a mass spectrometer that allows us to take a direct colony of bacteria, yeast, or mycobacteria and identify it to the species in a matter of seconds.
So when you say direct colony, do you mean you can actually take from a sample from a plate, let's say, that's growing?
Yeah, exactly.
And then you test it on this? Okay. So what does that entail? How do you do that?
So we have to get a good colony of growth, and then we'll take a small toothpick, take a little bit of our colony, and smear it on one of these plates. And we'll add a suspension of matrix, which is essentially just a layer of material that sort of protects the sample from the laser so that the proteins aren't denatured, so that they can be better analyzed. And so once the instrument shines the sample with a laser, it will cause the proteins to become ionized, and they'll travel through the instrument, separate based on size, and then that profile of proteins will be compared to a known database of proteins and will help us to identify what the species is.
Oh, wow. So how long does that take?
So each spot just takes a couple seconds. So it's greatly sped up our process from, we've gone to more of a molecular-based method as opposed to before when it was more based on biochemical characteristics and metabolism of bacteria, which we know can take 24 hours or more.
Wow. And this, because it's from a culture plate, it can be done on just about any specimen that we culture.
Oh, pretty much, as long as it grows in culture.
Okay.
Yeah.
And does the MALDI-TOF identify just bacteria, or can you also identify other organisms like fungi?
Yeah, so mainly we use it for bacterial identification, but we can also identify yeast and mycobacteria. And they're working on building the library so that someday we will be able to identify molds as well.
Wow.
Here is how it works.
Once inside the spectrometer, the target is brought into a vacuum and struck by a pulsed UV laser, usually at a wavelength of 337 nanometers. The laser energy is absorbed by the matrix molecules, which are energetically ablated from the surface of the sample, carrying the analyte molecules into the gas phase as well. During the ablation process, the analyte molecules are ionized, usually by proton transfer with the nearby matrix molecules. The most common MALDI ionization format is for analyte molecules to carry a single positive charge. Now that the gas phase analyte ions have been generated, their masses can be analyzed by the time of flight mass spectrometer. The time of flight process accelerates the gas phase ions in a high-voltage electric field. This field imparts a constant amount of kinetic energy on the ions. Because kinetic energy is equal to 1/2 mass velocity squared, a constant kinetic energy will cause the smallest of the ions to travel the fastest. In this example, we have a mixture of 3 macromolecules with masses of 1, 2, and 4 kilodaltons, where 1 kilodalton equals 1,000 atomic mass units. Using standard TOF conditions, this would cause the smaller 1 kilodalton ion to be accelerated to the fastest velocity, around 54 kilometers per second. The 2 kilodalton ion would be accelerated to around 38 kilometers per second, and the largest 4 kilodalton ion would be accelerated to the slowest velocity, around 27 kilometers per second. The TOF detector then records the time it takes for the groups of ions to travel a certain distance. From our previous examples, for a standard 1.5 meter flight, the smallest ion would reach the detector in around 28 microseconds, the next at around 39 microseconds, and the largest ion in around 56 microseconds. From these times, and the known constant kinetic energy of the electric field, the spectrometer is able to calculate and record the mass-to-charge ratio of each ion it detects.
So once it's finished, we'll get a report, and it'll have each organism listed with a score.
Okay.
And we'll compare the scores, and essentially what the score is a comparison of the ID to the database and how well they matched.
Okay. So this is a report of several wells?
Yeah, so this is our plate with a couple samples. Essentially all the ones that we're seeing that are green had a score above 2. And anything with a red score is a score below 1.6.
Okay.
And then anything with yellow is between like 1.6 and 2.0. And those are IDs that are okay, but they're not, they don't match to the database as well, so we can't report it to the species.
Okay. So, for example, this top one, A1, whatever that sample is, it's growing E. coli.
Exactly.
That's confirmed. Is this a confirmatory on this? There are 2.
Yeah, so it'll give us the top 2 scores, and essentially how it breaks down is anything that is underlined means there's something that's preventing it from getting us the ID to the species. So, for example, with E. coli, as we know, it's closely related to Shigella. And they're so closely related that we can't tell the difference on MALDI, so we have to back it up with a few biochemical tests to confirm that.
Okay.
So we use indole.
Okay, so this is E. coli or Shigella on MALDI.
Exactly.
Okay, and then down here, this one has Staphylococcus hemolyticus, and then that's a 2.13, but then right next to it is Staphylococcus, what is that? That's
hemolyticus as well.
The contacts aren't working very well, but it's 1.94 and it's yellow, so what does that mean?
So it just means that, so our top score matched as hemolyticus, and the next closest thing in the database was also hemolyticus, but the score wasn't as good.
Okay, so that's the second one. So, but you would feel comfortable saying it's most likely?
Yeah, and actually if we were in the program, we could look and see the top 10 and compare those.
Okay.
And so we don't have it in front of us, but there's a good chance they were also hemolyticus.
Okay,
So.
Wonderful.
Susceptibility testing is performed on the microscan.
So we've set up some saline prompts, which means we put some bug into some saline, and we're getting ready to do some susceptibility analysis. So Sam is going to show us how he inoculates an organism into the microtiter MIC tray. We have an organism already inoculated into this fluid, which is just some saline. He's now going to do a sterility plate or a check plate to ensure that we may ensure that the organism is sterile. He's going to pour the isolate into a seed trough. We want to get an even distribution of the organism. And now he's going to take a pipetter and he's going to uptake all the isolates and put it into the MIC microtiter plate. This ensures that we've gotten every volume, an equal amount of volume placed into each well. There are rows of antibiotics on here, various antibiotics, about 20 to 25 antibiotics, and they've been evenly distributed and inoculated. So Sam is now going to place the inoculated panel into the microscan instrument, which is an incubator and a reader. It will incubate overnight for 18 hours, and then the next day it will print out a readout of all the susceptible and resistant antibiotics.
So how do you know which patient that plate belongs to? Is there a
Everything
label
is
on there?
bar-coded. Yeah,
So that's
we have
also
barcodes.
barcoded? Okay.
Yes.
And you said that that incubates overnight. Does it beep also like the back tech? Or
No.
what happens? You just check it?
When it's ready, it automatically prints out. It does not autofile.
Oh.
We have the capability of autofiling it, but with the complexity of susceptibility testing these days, when we have susceptible and resistance, we want to make sure that we report it correctly, so we like the techs to review those results before they're released automatically. But if a physician would want to call us, we could always look it up ahead of time before we release it and let them know what's going on.
Okay, so it would show up in the chart, or it shows up for you all, or is there a printout and then you review it and then you
It prints out, we review the printout in the morning, and then we release it in the computer.
Okay, and you said it's about 24 hours, correct?
18 hours of incubation it
18
needs.
hours of incubation.
Right. For certain organisms, it needs a full 24 hours. For the Enterococcus, for the vancomycin, and for the Staph aureus, for the vancomycin. But the gram-negative rods are due in 18 hours. It doesn't give me anything before 18 hours.
Okay, and you wouldn't need to hold it much longer than that?
No.
So
That's standard protocol
so
for
leaving it
susceptibility
longer
testing.
is not going to give you any further
No.
information?
No.
Okay. And this is done for any cultures where we ask for susceptibility?
For any members of the Enterobacteriaceae, so organisms like E. coli, Proteus, Klebsiella, Citrobacter, Serratia, and more, and also all staph species, aureus, and all the coag-negative staph, as well as Enterococcus. That's probably the bulk of them.
Okay. So is there someone who runs the microscan for the week, or is it whoever works at a certain bench, like the blood bench or what have you, they prepare the microscan for their...
Each tech prepares their microscan on their corresponding benches, and then we have one or two techs throughout the day that run the microscan, but we rotate it. So
Okay.
every tech does have a handle on this
Okay.
and is able to do it.
And
Right.
so respiratory cultures also are done on
Yes,
this?
respiratory, wounds, blood cultures, aerobic wounds, anaerobic wounds, urines, all of them are run on here.
CSF?
Mm-hmm. Yes,
Okay.
that also.
Okay.
We also have the ability to perform rapid diagnostics for specific infections utilizing the Cepheid. What is very nice about Cepheid is that it allows us to screen for patients who may require isolation as specific carriers or for specific infections, and it helps us remove them from isolation sooner in the case of tuberculosis.
So we keep hearing about all these different tests, BioFire, MALDI-TOF, Cepheid. What is Cepheid?
So Cepheid is a rapid PCR instrument, and there are several different assays that can be run on it. There's C. diff, Group B strep, MRSA screen, the notorious MRSA screen. There's carbapenem resistant assays. There's rapid tuberculosis assays, and all of these assays come out with a result in about 1 hour. So it's very rapid.
Okay, so this can be used for screening, maybe
correct.
Taking someone off of TB isolation earlier.
Yep, that's correct.
And you said strep? Groupie strep. So we do
strep.
this for like pregnant women then?
Exactly. Yeah.
Okay, wonderful. So, so this is different from BioFire.
That's correct.
Okay, and it's really just, are you testing for one, you're doing a specific test for a specific
Yes.
identifier?
So this is more specific testing. Over there with BioFire, you had the syndromic panels with, 27 to 30 different targets, bacterial and viral, fungal as well. This is more of your specific 1 to 2 targets. MRSA versus methicillin susceptible, Staph aureus, things of that nature that just have one PCR target and just need the primers for that particular organism.
So you do this on what specimens? What types of specimens?
So the highest volume test is the MRSA swab, which is the red top swab here that all inpatients will get. Or I guess now it's been changed that there's an algorithm that only some patients get it. So that's just a swab of the nares. It comes in 2 swabs. The swab gets vortexed with a buffer, loaded onto a cartridge. Minimal hands-on time. It's less than 5 minutes. And then the cartridge comes over here and is loaded onto this enormous machine with about, I don't know how many, maybe 60 different doors that can be simultaneous running or simultaneous testing, and it's random access. Yeah, so that's pretty much what that is, and like I said, results in about 1 hour.
So does it beep or something prints out, or how do you know when it's ready?
So it actually auto-files. So like a C. diff. So let's say, for example, you have a stool sample that comes in. It's a C. diff that gets set up. It gets put in the cartridge, loaded onto the instrument. It does a PCR for 1 hour, and then it'll automatically go into the patient's EMR.
Oh.
Yes.
So you all don't have to do anything after
Nothing.
that, except maybe
Nothing.
empty?
Don't tell people that because we want to make it look like we do something. But yes, no, you're exactly right.
Wow.
And we actually have somebody who's going to set up a couple MRSA swabs here just as a demo.
Wonderful.
Okay, so here we have Indigo, and she has a MRSA swab, which is just a swab of the nares. So she's labeling the Cephia cartridge, and then she's going to take the swab and rub them together just to get any organism that may be on one swab but not the other. And then what she's going to do is break that swab off into a buffer that's needed for almost all PCR reactions. And then she's going to vortex that for 10 seconds just to really get any of that DNA, any of that cellular material off. No pressure. And then she's going to pipette that buffer combined with the DNA material into the cartridge. So it's very simple. It's not the PCR you're used to doing in college where you have to wait hours and hours and do all the sample prep and And then she'll come over here, scan the 2 barcodes, the patient barcode and the cartridge barcode, and then it goes on a belt and it gets loaded right in. And you'll see this gantry arm come over and take it and put it into its specific testing door. And once again, this is completely random access. So as the tests come in, they can be put on right away. So there's a lot of troubleshooting that's involved as well. But yeah, I mean, we're really diversified in our different instruments here. Some are very low tech. Some of our antigen testing, it's colorimetric. You're just looking to see if there's a colorimetric reaction. Some of it is actually, reading plates and determining colony counts, and then others is, setting up a PCR reaction. So it's a very diversified set of tests that we have here in the microbiology lab.
Wow,
Yeah.
that's incredible.
Absolutely.
And you just rotate different...
We do, yeah. Keep our brains fresh. That's what we like to do.
Very good.
Yes,
Thank
absolutely.
you very much, Sam.
Thank you. It's great to have you here.
Thank you.